We're going to share some of our progress in developing very precise single particle analytics, single cell analytics, so that we can help improve the next generation of these nanomedicines by taking an extremely detailed look at this fine level of resolution to better understand how they work and how to improve them. So as was shared, I'm a physicist, both in the physics department here and the Michael Smith Labs jointly appointed. And we develop new imaging approaches with the purpose of bringing new insights, shedding new eyes into these complex macromolecules. So the big picture vision of my research team is to really connect the dots from the level of the single molecule and its behavior, or the single particle, to the level of the single cell. And we work with health scientists so that we really focus on the therapeutic formulations that already have good contrast or evidence in that health sector. So the idea is, imagine you're a nanoparticle and carrying a cargo, the very last interaction between, the drug and the target. So it could be many kinds of drugs that you're interested in. It could be an antisense or an aptamer. We're going to hear today about many kinds. That very last molecular event is relevant. You might like to study the on and off kinetics, or the cleavage rate, or the steps of action. And with regard to the particle, following its journey, entering the cell, the rate of uptake, how many make it to the endosomal release, what happens to the RNA after that. So that kind of information is at such a granular level that we need to develop precise microscopy tools. So the background of this talk you may have seen before. We've developed a platform called CLIC, or convex lens induced confinement, which works by squeezing the molecules into a thin chamber while looking straight up through an objective. And we use entropic confinement, or avoid tethers, so that the particles or molecules are freely diffusing but isolated. So we can get quantitative information out of them without pinning them down, which would alter their behavior. And just as background, if you're curious, we've, like Peter, been very inspired to patent and enable our work to then be in the hands of industry to bring it to practice, with background in both the patent and academic space. If you'd like to learn more about these imaging methods at our labs, you're welcome to come by. Our background in starting out was very much focused on molecules, DNA, RNA studies, and with learning, especially during my sabbatical, part of the time I spent here, about the importance of the carrier of the RNA and other therapies into the cells, and lipids as the most important one from Peter. We've really recently focused on characterizing with great detail these single lipid particles and the RNA cargo, as well as their dynamics and moving into cells. So that is what this seminar is focused on, and I just wanted to provide a bit of context. So why find out this granular information? Well, it can impact the behavior of the formulation of your nanomedicine. So the size of the particle, we use PEG to optimize that size for cell uptake. It's good to know that in great detail. You might be preparing a batch of particles and watching their aggregation, or different populations with different sizes. And the reactions of the particles, either together, their fusion and storage, or their response to pH in cells, we'd like to study that in great detail. Not using the ensemble average tools like you might use in your labs right now, DLS, SPR, but in great detail where you see the whole distribution, and to follow the action. So that's what I'm going to talk about today. So just to help you understand the movies I'll show, we insert a sample prepared by Yao and other students sitting in the front row into a little glass flow cell, trap them, isolate them, and this is a little movie. The first one we took in Vancouver, actually, of labeled plasmids being trapped into wells so that we can watch them. And nanoparticles are about the same size as those plasmids. And we're avoiding tethers to really get at behavior in these complex, out of equilibrium dynamic environments. So that's an introduction to the background for this talk. In this talk, I'm going to quickly walk through a publication you might have seen online on first applying this click measurement platform to develop it with great rigor to characterize these single particles. And then new unpublished results on the dynamics of fusion of these particles, which we've done since relocating to UBC during the pandemic, which is a whole thing I could talk about. And they're beginning to look at their journeys in cells. And the kinds of questions we're motivated to ask, as I just shared, because they can help us improve medicines, are what are really those detailed distribution of size, structure, and relationship to potency. So the first step is to take a detailed look at the particles, trap them in individual wells, follow them in time. As you know, in developing new methods, very quantitative simulations with mathematical rigor need to be developed so that we can relate the outputs of these diffusing particles through mathematical analyses to their radius of gyration and therefore size. I won't get too much into that, but if you're interested in that, we develop a theory of confinement and diffusion and pulling out the diffusion coefficient. And we think that's a good measure of size in solution, complementary to others, because it's really reflecting how this complex, deforming particle, think about getting thermal kicks, it's also got lots of modes, how it moves. And that will be how things, biological cells sense it as well. So the first control we did, just to kind of ground this work, is to take a standard formulation, the Onpattro one. So I guess in Orr's slide at the far end, there was Orr's showed the first siRNA cargo therapeutic delivered in a lipid particle, which came before the mRNA vaccine, just in the rightmost end of his slide, released in 2018. So we were working with this one. We added a little bit of dye, just 5 dyes per particle, making sure there's no effect on dynamics or bulk measurements or cryo, so that every particle has a bit of fluorescent label. We track them, and then what's shown on this scatter plot you might think of as like a fingerprint for that particle's intensity and size characterization. So on the x-axis, if you look at the scatter plot on the right, you'll see that bigger particles are brighter, because as you go further in the log of the diameter and you go up in the log of the intensity, the brightness per particle, they lie on a slope. The slope is 2 because the dyes are, as we would expect here, in the single layer, the single laminar layer of the outside of the particle. So that was sort of a check. And then the spread on the log size axis gives you homogeneity or heterogeneity, and on the y-axis, the spread in the brightness. And you'll see that there's a big clump at the middle of that graph, indicating they're fairly homogeneous. And you'll see the cryo-EM pictures at the bottom of that graph, just below it, showing that they are indeed quite heterogeneous. So that's why these points are spread out on the size axis and the vertical axis. And then on the left, we took this Onpattro formulation, but looked at it in a different pH, as you'll see, pH 4. That's the pH where it's actually first prepared. These are prepared in acidic conditions because we're mixing the hydrophobic, hydrophilic ingredients. That's where you form the particle. And on the left, you'll notice just in cryo-EM, it looks a bit different, right? They look more bi- or multilaminar, and they're more heterogeneous. So now if we look up this scatter plot that we've taken, where every dot is a intensity and size of this tracked particle, you'll see that the distribution isn't perfectly along the line, but if you were to take an average slope of all those dots, where everyone's a particle, it's a bit steeper. And that's consistent with the multilaminar particle, because the dyes are now integrated more into the volume. If you had a uniformly filled volume, the intensity would scale as the radius cubed. And so this was a very good control for us to show that we have the single particle sensitivity and can get a size distribution and intensity distribution for every particle. Next, we stain the cargo. So can we count, 1 siRNA per particle? So when you shine a laser at a fluorescently stained cargo, the fluorescence will step off. It will photobleach. The laser no longer interacts with the dye. So if you label, say, just 1 siRNA in the particle, if you had the sensitivity to do it, you would expect, like what we see in that figure A, a single step. And then as you have more labeled siRNA, multiple steps. And typically for single, what's called photobleaching counting, you can quantify a few steps. So this is showing, can we count, number of labeled siRNA? These are fractionally labeled because there are actually hundreds or many siRNA in these particles. But the answer is yes. So this is developing the platform with the intention of really characterizing, what's more interesting, the mRNA. And then in the last bit of this paper, we actually did a blind experiment where folks over here in Peter's lab had some cryo-EM data that we had not seen. And this is taken by Jay Kulkarni and Dom Wisman. And it turned out that with the siRNA-loaded particles in cryo-EM, they were seeing nuggeted siRNA in the particles. So when we take these single particle measurements and assign each one a size and an intensity, we're no longer seeing dots along a nice correlated line, which would have indicated, it's not, for example, figure E, where they would have been on the surface, scaled as r squared, and it's not like figure F, where they might be uniformly through the particle. Then we'd see a nice scaling line on that scatter plot. Instead, it's just an uncorrelated blob. So we found that very mysterious, so I actually took a lot of data here, several thousand particles, to measure this uncorrelated blob. But it's consistent with the nuggets of siRNA, sort of bigger siRNA, smaller amount of RNA. You might have small amount of RNA or larger amount of RNA, as you see on the right. So it's a kind of fingerprint showing that's consistent with this clustering. Okay, so this is one piece of our journey to establish the single particle characterization. And then next, we were very interested to move, and this is now unpublished, to the same questions about the mRNA lipid particles as a function of formulation. So now we introduce double vision. We've well calibrated the staining of the lipid, about 5 dyes per particle, making sure no effect. So all particles are green on the left. And on the right, do they have mRNA or not? So if we stain the mRNA in a different dye and are very, very careful to do a lot of controls that the cryo-EM is not affected by that the long-term stability and dynamics, which you'll see we can also measure, is not affected by that, either in bulk or at the single particle level. So I'll just give a caveat that the choice of dye, and it's very important, these are very sensitive materials, as you'll hear about today. We can see how many are empty, how many are full, and I'm going to go that far today, but we can also count how many mRNA are in them. So on the right, if you have an empty particle, you can do very sensitive tracking in the lipid channel. On the left, if you have a full particle, you can use co-localization and very sensitive analysis to see the particle in the lipid and the RNA channel simultaneously and thus establish it is loaded. So now it's interesting to ask, and this is a good prelude to the talk by Miffy coming next, and is motivated by general interest in the field to get these quantitative analytics in place for asking and answering these kinds of questions, but also in parallel seeing such interesting results that structure does matter, loading and structure sort of play together, and this does relate to potency, so figuring out why and so on, building these tools is interesting. So the first thing we did here, and this is unpublished, is that we took the formulation as a baby step that I just showed, the Onpattro one with siRNA. Instead, we use a luciferase mRNA, and we modify the type of lipid just as a first step. And for those in the audience, and I guess all civilians now, mostly realize there's sort of some lipids that are being used in the commercialized vaccine by Pfizer or by Moderna. So these have chemical names up on this chart. But we're very interested to see, if just titrating those lipids makes a difference. So on the far right, and this is just a example formulation, I'm not saying that those are precisely the loaded fractions for the commercial product. I actually have reason to believe those are very, very high, at least for the Acutis BioNTech Pfizer formulation. But this is one where we have biophysically changed the amount and the which type of lipid with respect to our prior paper as a baby step here. So you will see that the red dots on these graphs are further to the right, so they're bigger and higher in intensity. Well, that's the RNA channel, so they're loaded. The blue ones are further to the left. They're smaller and unloaded. They've been seen in the lipid channel only. And if you look at the bottom, maybe the punchline of this, because it's a lot to share, is that the structures of the particles do seem to vary. Miffy is going to talk about her observations next, I think, of the bleb or acorn shapes that can occur where in cryo-EM one sees the mRNA sort of protruding like the head of an acorn or a bleb out of a particle. So we're seeing that here too in these cryo data taken by Yao in the facility across the street. And also you'll notice that as the size, I've posted here on the table the radius of gyration of the particle. I just want to say that that number is a bit bigger than what you might, if you drew a line on a cryo-EM picture, pull out. But they do relate. They're two different measurables. But bigger particles tend to, on average, have more RNA. And you'll see that the fraction loaded here is between 30 and 60%. The bigger ones have more mRNA. And that changing the lipids seems to change the structure from more acorn-like to not as acorn-like. I think that's a subject under investigation in how the mRNA structurally distributes. But this is really quite interesting. So we're also curious as we start to play with the amount of PEG, how things change. So one thing you'll notice right away is these loading fractions are lower and the average sizes are a bit smaller. So we're at the stage where as we vary formulation, we can differentiate those sizes and structures. And we again here in this perturbation of our formulation do still see these bleb and acorn shapes, which is, I guess, at the empirical stage of observation. And then next we were curious also to change we're fixing one of the formulations and varying the type of PEG. So PEG is something, as you develop nanomedicines, I guess folks are using to stabilize the size and in solution. And you can see again that this is having an impact. These loading numbers are a little bit higher, more like 50 or 66% compared to the previous one. And on the right, these bleb or acorn shapes are persisting. The loading fraction here with the Moderna one is a little higher. As we move to the next side, you see that this particular formulation has much lower loading and the particles are a little bit smaller, related to the fact that this large mRNA polymer probably no longer fits in the particle with this particular formulation. So I think it's interesting once we take a detailed look to see that small perturbations in the formulation have large change on the structure and quantitative loading, and we now have the step where we can follow these particles and count. Or here I'm showing empty and full, but by the photobleaching method with singly labeled mRNA, we can count. Here I've shown data, if you remember, with a few dyes per mRNA. Just in our first data, we wanted to make absolutely sure we didn't miss anything due to photobleaching, so we have a few dyes per mRNA. So that's a recent contribution as part of our bigger picture. Our bigger picture is, of course, as we try to break things down in pieces to be able to track particles, see size and loading, see the dynamics, follow them in the steps inside the cell, and relate all of this to try and really optimize and understand what's occurring. So one of the things that we got really curious about, especially in our controls, where we saw such different structures at pH 4 and 7.4, and relating back to my comment on manufacturing is, what are the dynamics in between? And a part of what motivated that is we're making the samples in the NMIN facility in Peter's lab. And we noticed that if we store them for different times or wait different times in the fridge or walk maybe different times, you could get different results. And maybe that's a really important thing to study because in the field, one of the gaps is perhaps really making rigorous the properties of these particles in many labs, in many places to help move the field forward. The most quantitation we can contribute, the better the field can move forward. So we thought it would be really interesting from that practical perspective and also biophysically to look at the dynamics of the particles. So this is one experiment, and of course is also to develop this because we know there's so many other dynamics and interactions. These are like out of equilibrium to study. So in this particular study, which we're just submitting now, we have half our particles. We're doing it with the Onpattro formulation, so moving back to the first part of my talk, labeled in green and red. We load them in a click device, but we take advantage of this entropic confinement. Ooh, this is what happens when you transfer your talk to a memory stick. And oh well. Okay, so basically, yeah, at short times, you would expect particles to diffuse independently. And then we change the pH. We go from the acidic to neutral pH. And we actually see them fuse. And so we've been very curious to quantitate that. So this is a larger frame of many of these multi-particle wells. I'm showing overlaid in green, where we shine green excitation and get green light out of the single particles. And in red, we actually see this signal called fret when they fuse. Now, this is a little sidestep on the optics if you're not familiar. With red and green dyes, if you shine green light, they can actually excite, the green emission can excite the particles label in the red to get a signal called fret. So here we actually see for every well, which we can crop out, the fusion reaction, and we can get a time course for every single one and pull out the rate, the amplitude of the signal, which does relate to size of the fused nugget. So very quickly, we were curious to study this fusion. So this is occurring in kind of a phase space between the initial particles at the initial buffer and pH and the final condition. And so we took different paths in that phase space. We went from a different initial to final buffer, in other words. And the structures are quite different at the start and the end, right? And there's a lot of questions about how this impacts potency, as you'll see through today. So this is taking a detailed look at those particular fusion that would occur after or during transport from the manufacturing buffer to the storage buffer, but biophysically quite interesting. And I don't have a lot of time, but I just want to say the main point, which is that pH, we took different paths to now study ionic strength and pH to see what really drives this fusion. pH seems to dominate. The timescales can vary from days, if you look at the time constant of those red dots, to seconds, if you look at the time constant of those purple dots, depending on like which buffer you switch. So maybe you can come visit our poster to learn more about that. But you can see that the structures in cryo-EM at different points are actually quite different as well. So it's very interesting to think about also what's happening in between. You start with one structure, you move to another, sort of scrambled in between because they're switching from one configuration to another. And from our perspective of nanomedicines, the configuration can influence potency. We care about stability. We care about dynamics. There's an optimum. And inside the cell, the pH is dropping in the endosome, which is the opposite path of here. But it's really important to understand this as we look at results in cells. So we asked some questions here about how does the payload or the cargo ratio impact these time scales. And very briefly, you see the dots on the right. That was that diagonal curve that had sort of the fastest. We can see that as we have a higher N/P ratio or more loading, we basically slow down the fusion. And we asked, how does PEG impact this? So we also saw a general trend, so scatter plots for every condition of PEG variation, and where each color is the path we talked about before, where varying the PEG results in a lower fusion activity. So there's some clear outcomes of this work where we see a clear correlation between formulation, structure, and fusion. And ORS ran over time, so I guess just stay on schedule, yes. Okay, so what's happening now in my lab is, thanks to NMIN, I'm pointing at Diana Royce, who's speaking later, and others, we have these assays working. We're taking more and more measurements with members of Peter Kull's lab and my students teaming up to really respond to therapeutic observations at the level of mice and formulations, and you'll hear from Colin and others in the NMIN Grand Challenge today, and take a detailed look. So that's working, and we've also been able to upgrade some microscopes to really focus on that, so that instead of one, we'll have 3 able to take this data very, very soon, with the help of also engineers at the BC Cancer who have scaled up things like sequencing assays. And in addition, thanks to the CFI and this community and the province of BC, we have a couple new microscopes that are so exciting because they allow us to really push the limit and use the combined technology we've developed for confinement. So that provides a lot of insights in terms of connecting and analyzing those detailed trajectories as they follow cells to what we see in terms of formulation and structure of the analyte. So we have a couple of these single-cell microscopes set up. And if that wasn't enough, we're also super curious to always benchmark our results You might think single particle, single cell, why do you care? Well, from the genomics community and screening groups especially, just came back from Sweden where I was asked to speak at AstraZeneca. It is established in the field correlation between single cell observations and therapeutic results at a macro scale. I visited a facility with 3 floors of mass spec, genomics, microscopes. I asked, well, what can I do? But they think this is important, so that's good. But we would like to cell by cell also sequence and look at proteomics and follow the individual trajectories of the particles. So that's something we're working towards. So in terms of the single cell imaging, what we're doing right now is we're using the very same formulations as a starting point, where we label the RNA and lipid. You can put fractionally labeled materials into the cells so that you're not altering all the results, but you follow the labeled ones. And yeah, ask questions like how many particles are entering the cell, how many make it to the endosome, how many are there as the pH changes, because there are dyes that respond as the pH drops, how many make it out, how many RNA make it to the ribosome. So very quantitatively, and I suppose one way to put this is, I'm not sure if this figure is correct, but Pfizer or other pharmaceutical firms made, I'll just say tens of billions of dollars in the pandemic, but their product is 1% efficient. So, hopefully the physicists in our teams can, make these products better, reach more patients, and as you hear about in Peter's talk later, optimize these for many kinds of diseases. So I'll just stop there. The single cell results are fairly new. If you'd like to visit, that's where we're really driving. And I am at 28 minutes in my talk, so it'd be great to accept a couple minutes of questions. These are, the names of the trainees who have worked on this particular project in one of my subgroups. Yao is here, and collaborators in particular from Peter's group, Miffy, Bin, and Jerry, I think, are here. Some of my students, Eric, postdocs, Albert, and Ari, may come by later at the posters, and we have poster 21 and 22. I was also just going to announce we have a few positions available, both starting immediately, but also in the spring. So if you're inspired by this or want to participate in any way, please also let me know. And huge thanks to this community and Min for the opportunity to give this talk. Thanks. I guess we have time for like one or two questions.
One or two questions.
Just any burning questions from maybe a student? Oh, great. Terry, the best student.
And it's very elegant what you're doing, but the thing that I wonder about is what happens when you go in vivo? You've got so much more complexity, things bumping into other cells, things leaving plasma, plasma oxidants, stuff falling off of the zones, whatever. I mean, are you able to start to deal with those kinds of complexities going from in vitro to in vivo?
Yeah, and I think that's really the question and the purpose of the work. So I guess the approach we're taking right now is that we're working on formulations, in these controls we've obviously made perturbations away from those, but where that in vivo and clinical data has already been taken. And I suppose one way to answer the question is preliminary screenings do show correlation, I timed myself as well, between at least single cell and upwards. And then this characterization is kind of about why, the mechanism of action. So the world is also moving towards sort of big data AI approach, so there'd be characterizations of toxicity, entry, getting to the right cells, and then this result would fit at the single cell level and the level of the analyte and kind of the why. So I think the answer is one would hope that this information would help you sort of do a lot less of those complicated experiments later on, save the mice and be more intelligent about it. The other important intermediate thing, I guess, we have molecule particle cell. We can do tissue and organoid if we have 3D scanning, but really over in life science, there's some platforms focused on organoid. But I think the hypothesis is yes, that this will help, but it's like part of a funnel. So it'll probably be the case you'll have huge screening, less screening, and then your best ones, or comparing your best and worst ones, single cell and down, because that's where the molecules take their last step. But then that will help refine the future broader screenings one. So I think that's how I see it. You wouldn't want to do this in a vacuum, but it's not. I think its current role is to understand why some work and not, and then as things evolve, it'll probably become increasingly predictive. And I've learned in travels and just interactions that certainly this kind of data is being used elsewhere and does correlate. But of course, some of that's not even published. You wouldn't necessarily know. But it's a great question. But I guess I'm a physicist, and there's the hypothesis that the atoms and molecules are important to predict what happens. But there's several layers of information to use.